Thank you. So I'm Allie. I'm a senior. I'm graduating in May, with a degree in chemistry and a minor in biology, and then I'll be going on to the University of Utah for my PhD in immunology and microbiology. Thank you. So this is data that's going to be presented not just at Research Day, but also in Chicago at the American Association of Immunologists annual conference. So my work is on expression and function of Fc-epsilon-R1 in breast cancer cells. So during this presentation, I'm going to show you all the data for my human cells, not the mouse cells. I'll leave that for Research Day. So a little bit of background. I am working with triple negative breast cancer. So what triple negative means is that it has no targetable hormone receptors inside or outside of the cell. So we can't use traditional hormone therapies to treat this type of cancer. So your only options are chemotherapy and surgery. So I'm kind of looking at particular immune cells within the cancer itself, as we can use those to treat the cancer instead of targeting receptors on the outside of the cell, of the cancer cell, because we can't do that. So I'm looking at a mast cell, which is this guy here. And what I'm looking at is this purple receptor here called Fc-epsilon-R1. And it binds to IgE, which is an antibody produced by B cells for specific antigens. So if you ever had an allergy, you ever have anaphylaxis, that is your B cells producing IgE for an antigen to peanuts. That's what this whole mechanism here is. So once antigen binds to IgE, then we have what's called degranulation, which is a release of histamine. Histamine is what causes you to have anaphylaxis if it's released in large amounts. So my methods that I use to collect all of my data, I use confocal microscopy, which is basically a really fancy microscope that uses immunofluorescence on tissue to show me the specific receptors. So I looked at Fc-epsilon-R1 on tissue sections. I also used flow cytometry, which is a more, so confocal is more of a qualitative type of analysis and data collection. Flow cytometry is essentially the same thing, but it's quantitative. So I'm getting numbers for the tissue samples that I used. And then I also did several ELISAs. ELISAs are enzyme-linked immunosorbent assays. So I basically put a bunch of IgE and antigen onto these cells, and then I measured an immunoglobulin that is released because of signal transduction. So the downward effects of that receptor being activated, I measured those like things that are released. And then the last thing I did was, we'll get this receptor is present. We need to find out if all of it is present, because receptors aren't just one part, the thing that sticks out of the cell. It's multiple different proteins. So we looked at that to see if all of the subunits are present within these cells. So my results. So this is the only mouse data I'm going to show you, because it's really hard to get your hands on human tissue. So I couldn't do this kind of stuff with human tissue. So these are all confocal images. So up here, these are wild type controls. And the green, that's the Fc-epsilon-R1. The blue, that's all the nuclei of the cell. So, and then down here are IL-6 knockouts. So IL-6 is a pro-inflammatory cytokine that is released by mast cells once Fc-epsilon-R1 has been activated. So we removed IL-6 because if you remove a pro-inflammatory factor, inflammation goes down. Once inflammation goes down, in theory, the tumor should have less of an inflamed area to expand, because inflammation is not good for your regular cells, but it's really, really good for cancer. What we found is that Fc-epsilon-R1 is upregulated, which is the exact opposite of what we wanted to see. So it's like the jumping off point for my research. And so I was like, well, this is weird. So let me do some flow and see like quantitatively what the expression is like. So at the top, these are all unstained controls. Bottom, this is stained for FC epsilon R1. These are our mast cells. This is our positive control. And then we have 3 different human cell lengths. So these were EWD8s. So EWD8s are a more aggressive form. These are our triple negative cells. We also have 2 luminol type cells, so they have estrogen receptors. So these are E3s and MCF7s. So as you can see, the less aggressive cancers are FC epsilon R1 is not as expressed like in large amounts, but our triple negative are similar expression to our mast, which is a really interesting find. So if, in theory, if we can find this in natural human tissue, then we can use FC epsilon R1 as a target for cancer. So then we looked at functionality. So if this receptor is there, we gotta make sure that it works, right? Because that's how you make a treatment work, is the receptor has to be working. So this receptor is calcium dependent, which means that when it is activated, calcium is released from the endoplasmic reticulum. So these are our mast cells, and then this orange line here is our unactivated, just have IgE on them, and then we added antigen. And then you can see calcium goes up. So that's a normal mast cell. That's what we want to see. But our 3 cancer lines, nothing really changed. Very, very linear. Or even went down. So that tells me that it's either calcium independent or this receptor is non-functional. Gotta do a little bit more to see functionality. So we were like, well, it binds to FC epsilon R1 binds to IgE. So I should look at what IgE is doing for the growth. Nothing. IgE has no effect on growth of these cells. I didn't use LAD2s, which are the mast cells, because they're very slow growing cell line that I would base it. I would get a linear line here. There would be nothing to show. But these things grow like weeds, so it was very easy to plot a proliferation rate with these guys. And then I did PCR. So PCR is polymerase chain reaction. So I looked at the DNA or the mRNA present in each one of these cell lines, looking for each subunit of the receptor. So this receptor has 3 subunits, an alpha, beta, and a gamma. The beta and the gamma are actually inside the cell, within the membrane, and then inside the cytosol of the cell. And they are once IgE binds, the alpha subunit changes shape and kind of moves the beta and gamma subunits to cause signal transduction downstream of the receptor. So each one of these subunits is critical for function. And E3s, EWD8s had all 3 subunits. GAPDH is a loading control, so it's a highly conserved protein that should be in equal amounts in every single cell. So that's what that is, and as you can see, it's fairly similar across each cell line, which is what we expected to see. So future directions. The one thing I didn't show here is my IL-6 ELISA, because the last one I didn't work, so I have to do it again tomorrow. But basically what we're seeing is that these cancer cells don't produce IL-6. So the thing with this is that it has all 3 subunits, but it means that further down the stream of signal transduction, something is different. That is causing other things to be made or leading to this receptor not being functioning. So right now our conclusions are that this receptor is non-functional. It is just a fragment that has been created from these cells being immortalized, because this was all done in immortalized cell lines. And preliminary work that I've done on actual human tissue through a genomic database called R2 has shown that this receptor is actually downregulated in tumors. So this is a cautionary tale for scientists working with these cell lines to be aware that there are a bunch of different mutations that we don't know about, and this is one of them. Thank you.
Question for Abby.
Yes.
You mentioned calcium, like really brief. What does that have to do with your second?
Yeah. So this receptor is calcium dependent. So basically what that means is that once it's activated, signal transduction occurs and calcium in your cells is stored in the smooth endoplasmic reticulum. And that's where this downstream effect ends, is at the endoplasmic reticulum. So it's kind of like your muscles. Because like that's what happens to your muscles, is like when you have, and with your nerves as well, goes downstream, and you get a huge release of calcium. That's like, and it's basically an electrical signal. That's what's happening here. It basically is triggering, that calcium release is triggering that cell to degranulate. That's what it does. That's what calcium does in traditional mast cells.
Can I ask what like brought you to this research? Because I mean, this is an amazing topic.
So what I did was I was on the UNC biology faculty page, and I was scrolling through all the faculty. I was reading through all their research, and I came across Dr. Poland, and I was like, this sounds interesting. So I sent him an email, and he was like, yeah, I got a project for you. So he gave me this project, and he was like, yeah, so I had this master's student, right? And she got all of these tumors. I want you to stain them and see what we get. And so this was like the actual like first thing I did in the lab. He was like, here's this $500,000 microscope. I don't know how to use it, figure it out. Quite literally that's how that went. And I collected these images. And so I presented this at a lab meeting, and because originally I was just going to work with these tumor samples, I wasn't really gonna do a whole lot more, but they were like, this is really weird. This is really green. It should not be this green. And so they're just like, do flow, do this, do that. And so my project evolved from just working with a mast cell to working with a receptor. So I went from a large thing, which is cell, to a very small thing.
You hope to continue doing this type of work or even this type of project into your PhD program?
So I want to continue working in cancer because I've already done a literature review. That's 20 pages, and I kind of want to keep it. Because that was 3 years of my life working on that. And the PI that I've talked to that I really want to work with, because I am going into rotations, so I have to work in 3 different labs before I decide who I'm going to work with, wants to work in cancer and specifically wants to work in drug development, which is like kind of the direction of this project, was like to eventually get to drug development. But honestly, with like the results that I'm seeing, I don't think that's going to be plausible. But working with other immune cells, because mast cells aren't the only immune cell that are tissue residents. Like we have dendritic cells, we have macrophages. So maybe looking at a different cell line that could be used as a target, like maybe even developing like a CAR T cell type therapy, which is actually just been FDA approved, CAR T cell therapy for cancer. Maybe trying to detect like a specific receptor that is unique on human breast cancer cells and develop a T cell that can target that cancer. That kind of be like the direction I want to go down. Any other questions?
I just have a quick question about what it means to be an immortalized sample or
Cell line?
Does that happen?
Okay, so an immortalized cell line is basically, I mean, obviously these cells die, they divide and then they die, but they can divide more than once before they die. So for that reason, they grow exponentially and not linearly. So an immortalized cell line is a cell line that also does not need blood or heat. I mean, it needs heat, like we keep them in an incubator, but if they're frozen, we can unfreeze them and they're still alive. It's also a situation where like we just put them in a glucose amino acid lipid solution and they exponentially proliferate. Like that's what an immortalized cell line is. It doesn't die.
I can see how that would be somewhat different from ordinary.
Yes.
Thanks. All right, thank you so much.